Hello guys, welcome to my videos. In today's video, we are going to see every MacLeod and McCarthy experiment. We have seen Griffith's experiment in detail, and the complete video on this experiment has been already uploaded on my YouTube channel. Please do check out. In Griffith's experiment, we have seen that something factor or some factor is transforming from one cell to another cell according to the Griffith, which was named as transforming principle. He used in vivo conditions, that means he used strain Streptococcus pneumoniae and lab animal as a mice. But every MacLeod and McCarthy used in vitro conditions to continue the work of Griffith. They had used virulent strains of Streptococcus pneumoniae which were grown on blood agar to check transformation is occurring or not rather than using mice. So let's see the experiment. As you can see in this diagram, or we know that the hypothesis was the genetic material of the cell is either protein or any nucleic acid, that means DNA or RNA. So many scientists were considering that proteins was the genetic material, but after the work of Griffith, every MacLeod and McCarthy continued the work of him, and they used heat killed S cells. First of all, the whatever type of type 3 S strain was there of Streptococcus pneumoniae, it was heat killed by high temperature. Then the solution of this heat killed cell was prepared, and from this solution lipids and the carbohydrates were removed, because we have to check either protein or DNA or RNA is a are nucleic or are genetic material or not. Thus lipids and carbohydrates were removed. Then there three separate solutions were made. In first solution, proteinase enzyme was added to degrade or digest protein. In second solution, RNase enzyme was added to digest RNA. In third solution, DNase enzyme was added to degrade DNA. Then these three separate solutions were mixed with the culture medium containing live R cells. Then they conclude or they observe the results as in the first solution S cells were appeared according to the Griffith. That means some transforming factor was transferred from dead heat killed S cells to R cells to become S cells, we have seen already. So in first solution S cells were appeared. In second solution, in second culture medium, similarly S cells appeared because R cells were transformed into S cells. But in the third medium, R cells were not converted into the S cell, thus no S cells appeared. Why? Because transformation requires DNA. Therefore, it is the genetic material of the cell, has been concluded by every MacLeod and McCarthy. So what was the conclusion? We can say these experiments determine which molecule of S strain transform R2 strain into S3 strain. That means transform molecule is nothing but the DNA. So it was the first experiment which conclude that DNA as a genetic material. And final conclusion was that the culture which was treated with DNAs, as you can see in the third tube, did not yield transform type 3S strain bacteria, which indicated that DNA was the hereditary material which was responsible for the transformation. So this was the every MacLeod and McCarthy experiment, which was shown that DNA act as a genetic material. So thanks for watching.